Hello, my name is Stephanie Widman. Today I'll be showing you some practical staining strategies for improving your marker's resolution. Ever wonder why some scientists' flow data look so good? It may be because staining protocols have variations. When the technical aspects of these variations are explored and applied appropriately, they can improve a reagent's resolution, resulting in better resolution of your populations of interest. The simplest way to increase the resolution of your markers is to 1. Increase the signal, the positive MFI. 2. Decrease the background signal, the negative MFI. Or 3. Decrease the negative spread, calculated as RSD. These are the values used to calculate stain index, which is a measure of resolution. If you are able to do all three, that's even better. First, make sure you are starting with the correct staining protocol. There are many protocols available, and they include steps or conditions specific for the cells or application you are using. Let's explore some of the conditions that vary between protocols, such as temperature, staining volume, incubation time, and the use of BD Horizon Brilliant Stain Buffer. There may be a time to give markers special treatment. For example, chemokine receptors undergo a basal level recycling that is enhanced by the binding of their appropriate ligand. This means they may benefit from a pre-incubation at 37 degrees before you add the rest of the markers in the panel. Here we see human PBMC stained for CXCR3 and CCR6 chemokine receptors. The left set is stained per normal protocol of 30 minutes at room temperature. The plot on the right has an extra step of pre-incubating the chemokine receptor reagents for 10 minutes at 37 degrees. It may not seem like much of an increase, but when combined with other factors, such as age of blood, fresh versus frozen cells, this bump in signal will be appreciated. One thing you'll notice in different protocols is that the incubation times have a pretty wide range, anywhere from 15 to 60 minutes. Due to the effect of temperature, volume, or cell concentration, it may be best to stain your cells for longer. Here you see the difference incubation time has on the positive MFI value. Keep in mind that if you stain for a short period of time, your staining may not have reached maximum binding. This could add variation in resolution between experiments. Not all antibodies bind at the same rate, and some markers benefit more from the longer incubation. Staining volume is important to consider in staining protocols. For human whole blood or prepared PBMCs, 100 microliter cell suspension plus antibody cocktail often reaches volumes between 150 and 200 microliters. You can see in this graph that as you increase the volume of the cocktail, you may affect the positive MFI, giving a lower stain index or lower resolution. Due to this effect, it's best to incubate your titration experiments or single color reagent performance tests at the same volume as the panel. This way, you ensure that the resolution of the panel will be similar and there shouldn't be any unpleasant surprises. An important step in either mouse or human staining protocols is the need for washing. For whole blood staining, it's recommended to do at least 2 full washes after lysing buffer has been removed. Here you can see the calculated increase in stain index with each additional wash. Typically, this kind of increase is from tightening up of the negative cells and decreasing the negative MFI. Remember, the negative cell values are important when calculating stain index. Of course, there may be times additional washes are not possible, such as when you have extremely low cell counts. But if you have enough happy, viable cells, the additional wash may increase your marker's resolution. Lastly, when the use of BD Horizon Brilliant Stain Buffer is recommended, BSB and BSB Plus are staining buffers designed to mitigate the dye-dye interaction that occurs between polymer dye reagents, such as the BD Horizon Brilliant reagents. Best practice is to use 1 test size per tube or well. It is crucial to add BSB first, before the Brilliant reagents. If you're using BSB in your panel, remember to include it in your controls as well. It is best practice to treat your controls the same as the panel. As you can see, there are many aspects of staining your cells that may affect your marker's resolution. Alone, the increases may be nominal, but taken together, you may see significant improvement in your data. For more information about the products or concepts featured on this video, please visit bdbiosciences.com.